I'm Fiona Greer, and I'd like to welcome you to the second in our series of short, informal roundtable discussions on the theme of biosimilar analytics, hosted by Biopharmaspec. The topic for this discussion is knowledge-based biologics development and its implications for biosimilars. Today, I'm joined by two experts, Dr. Andrew Reason, an analytical characterization scientist, and Dr. Ash Ramzan, a regulatory professional, to give their views on why it's important to understand the ultimate regulatory expectations at the very start of any product development characterization, even early stage, to ensure smooth passage through into the clinic. Andrew is founder, CEO and MD of Biopharmaspec. He has 25 years of experience in analysis of both novel and biosimilar biopharmaceuticals and has been involved in the commercialization of a number of analytical methods for characterizing proteins. Andrew has contributed to many industry publications and is a regular presenter at conferences. He is also currently a visiting professor at the University of Warwick and recently joined the Macquarie University Scientific Advisory Board. Ash is founder and principal consultant of Woodley Bioreg, a global regulatory consultancy. He has a specialist interest in biologics, biosimilars and vaccines, and is a member of the British Pharmacopoeia Working Party, advising the government on the development of biosimilar standards. With over 30 years of hands-on expertise, Ash continues to work extensively in developing regulatory strategies for global registration of biologics and biosimilars, including MABs, vaccines and toxins. Ash is also a fellow of TOPRA, as well as serving on their editorial board. So thank you both for joining me today to talk about knowledge-based biologic development. I'm interested to hear how you define this concept and how it may be applied in the biosimilars sphere. So my first question to Ash is, what from a regulatory perspective is knowledge-based biologics development?
Thank you, Fiona. Andrew, hello, good to see you again.
Hi, Ash. It's been a while, but it's good to see you.
Yeah, it's an interesting question, Fiona, because as far as the regulations are concerned, there isn't an actual definition of knowledge-based development of anything. It's an intrinsic part of the way products are developed. But if we think about biologics and biosimilars, they are intrinsically complex molecules, and by analysing and understanding their structure, function, you are gaining knowledge in how these molecules are put together, their primary, secondary, tertiary structure, any post-translational modifications. But if you think back to the good old, O-level, A-level chemistry, biochemistry days, epitopes, how they're formed, they form a critical part. So what our interpretation from a regulatory standpoint is, as we gain knowledge of these molecules, the expectation is that you start to apply this knowledge in future developments or future iterations of these molecules, but also new molecules that are in the pipeline being developed. I can add, I mean, this isn't just about in terms of accelerating their development, but it's also about mitigating risk. So if you know a certain type of change or structural conformation presents a risk to patients in the future, then you steer the development away from that. And again, the regulators don't stipulate what you must do, but the expectation is that you use existing knowledge to accelerate through an iterative approach.
I see. So it's historical product knowledge that you're utilising to be able to move forward.
Well, remember that historical knowledge is something that you're currently doing, because as soon as it's done, it becomes historical. So it's that live understanding, interpretation and building using the experience and knowledge, again, of these complex molecules. Yeah. The predictive models aren't as simple as small molecules where it's A plus B equals C. They behave in their own way, and it's up to us as researchers, regulators, scientists to try and unpick the way that they work.
Okay, all right. In that case, Andrew, how does a knowledge-based approach then help with structural characterization and comparability assessment of biosimilars?
Well, yeah, I mean, it cascades down from the product, as Ash is saying, really. I mean, if you have some knowledge about the product, a good example, I guess, is antibodies. You know, antibodies are being developed in lots of different laboratories. Within the different subtypes, you know, there's a lot of knowledge exchange. You know, if you're working on one IgG1 and then move to another, there's a lot of transferable knowledge there. One of the things I think, though, that's forgotten a lot is the product isn't just the active ingredient. The product has excipients, you know, that it has things that might interfere with the analytics, and you know, that knowledge is huge in terms of time, cost, efficiency of designing your methods, you know, so that they are giving you the information that that you require. Good quality information then needs to be interpreted properly. Obviously, you know, you don't want to over conclude, so there is a knowledge base in taking whatever data that you have generated from an applicable method, put it into context for that product. So, you know, knowledge goes all the way across from the product itself, the excipients, to the methods. You know, what are the best methods for that particular product type? Are there excipients present that may interfere, that need to be removed? And then getting your data, and you know, orthogonality is a big thing from the regulator as well. Getting data, assessing certain structural features from different angles, and the knowledge in taking all that data, putting it together to give a valuable resource for that particular product. You know, having a knowledge base that starts that process creates time and cost efficiency.
Yes, yeah.
If I could just chip in there, Andrew, I think I agree, because the methods, the analytics you use will potentially be used in the future when the regulators come back and say, actually, we want you to do this test, and this test as part of demonstrating whatever they're interested in looking at. So having that solid analytical base is absolutely critical in future developments for next generation iterations of products.
Yeah, exactly.
On that, Ash, given that modern methods of characterization can interrogate the primary and higher order levels of structure and integrate this knowledge with the biological functional information, then from a regulatory standpoint, what are your views on clinical predictability based on analytical characterization?
Wow.
That's a big question.
It's a really big question. So there are a number of considerations that we can look at, and there have been some changes probably from the late 80s, 90s onwards that have subtly guided us into what potentially could happen in the future. So I'm old enough to remember comparability protocols and well specified biologic as it is now, and it was well characterized at the time. Now, those regulations were designed to enable the determination through a structured approach of structure function, so primary, secondary, tertiary, glycosylation, sugar groups, lipid, anything else that was in the product. And Andrew, you raise a valid point about the product isn't just the 3D structure, it's everything else that we put in there. So, and following on from that, we had comparability protocols. So the comparability protocol is primarily designed to use biochemical bridging to enable changes in process, product, formulation, you know, sometimes quite drastic changes, even sometimes cell bank changes. So there's already an understanding within the regulators that there's going to be a certain degree of negotiation when it comes to being able to downgrade the level of clinical data for these types of changes. Now, whether that interprets into clinical predictability is a really difficult question, because it's a 3D structure that is the effector molecule. So a subtle change in a conformation may render the molecule completely inactive, or indeed it may render it to be completely toxic. I mean, this is an unrelated example, but if you think about thalidomide, it was the L and the R form and it was the L form that was the teratogen, and it was the R form that actually had the beneficial effects. Now the R form now is being used to treat multiple myeloma. So it comes back to knowledge based, but the clinical predictability, I think there's always going to be an element of some clinical studies. Yes,
I think undoubtedly, yes, yeah. Andrew, do you have any comment regarding structural predictability and the importance of linking the structure and function?
Yeah, I mean, I asked the same question that you've just asked at an antibody congress about 10 years ago, Fiona, and there were experts from some of the largest biopharma companies in the world, and nobody had an answer to it. And I think if I probably asked the same question tomorrow, it would be the same answer. You know, antibodies is perhaps one area where you could see, you know, structure being related to function or binding or cell recruitment or whatever it might be, because there's lots of knowledge about the FAB and the FC regions. And that's kind of where my question was, you know, do we know enough to be able to put these things together with some sort of functional and then clinical expectation? To an extent, the functional thing, I think, has been done, but clinically, you know, we could talk about Northwood Park and T-cell recruitment and all sorts of things. You know, clinical predictability is very, very difficult. And, you know, structure and function are entwined. You know, they go together and indelibly linked, and, you know, move in both directions. You know, structurally, if you see something that is different in a significant sense, you'd want to know, are they functionally comparable? Therefore, does that structural change matter in terms of the true functionality of that molecule? Or if you have a functional assessment where, you know, there is a difference in binding or response, you want to know what the structural base of that is. So you can go back to the process and control that and you know, wheedle that out of future batches. So structure function, I think there always will be a link, and there's a lot of knowledge that goes both ways, but clinical predictability from structure is, you know, all sorts of things happen, but, you know, you wouldn't predict on a daily basis in clinical trials, and I think we're away from that at the moment.
All right, thank you.
It's worth pointing out that, you know, even in clinical studies, there's certain levels of unpredictability in those studies because, you know, ultimately it's people and they're intrinsically variable. People are not the same one to another. So yes, there is, but it's a really interesting question, and I hope one that someone in maybe 10 years' time, Andrew, can answer.
All right, well, we'll move on to our third question. As you both emphasised, structural studies are the foundation of biosimilar development. How can the recent movement towards analytical quality by design help with the structural characterization process? Is this something that you're seeing starting to be adopted, Andrew?
Yeah, yes, we are. I mean, I'm just going to share a slide. For me, I think if you were setting up and developing and optimizing your methods in the correct way, nothing really has changed in terms of what's expected in analytical QBD. You know, really the focus is on strong assessment of your method through the development of it, so you understand it very well, and that really then cuts down on, you know, changes, things happening that you're not expecting, or having to requalify and revalidate on a you know, a fairly regular basis. So analytical quality by design is really set in the method development phase. And the new ICH guidelines, Q2 revision 2, are focusing on the method development phase and the robustness of the method quite significantly too. Perhaps we can come back to that a bit later, but basically, you know, the way we would develop methods, the way we always have, and I remember having a conversation with Ash about this about 25 years ago, but we would always try and understand everything about the method, throw everything at it, look at all the variables, and out from that would come, you know, a very reliable, very strong method. We've taken that on in biopharma spec, and the bottom part of the slide is really where we get to after we've been through the method development phase and perhaps qualified a method. Because the other important thing to realize about QBD qualifying methods, you're not just qualifying it to show that it's reliable, you're qualifying it to get some understanding of the experimental variation of the method, because pretty much everything we do analytically is really a comparison. You know, it's not an absolute definition of structure. Does this match look like this one? Does this biosimilar look like that innovator? And to be able to call whether something is comparable or similar or not, you need obviously to know what is the inherent experimental variation of the method. And I think that's the other important aspect that isn't mentioned in detail, but comes out of setting out methods properly with all the background information. And the bottom row is just an example of our peptide mapping qualification process, where we have full knowledge of experimental error. We set up a very strong SST, system suitability test, using the qualification mix, so that we can show, you know, every time that the instrument is valid, qualified, reliable for use, but also defines the expected experimental variation.
Well, thank you, Andrew, for sharing all this amount of detail from your own laboratory experiences. That's terrific.
Well, let me first of all just say that, Andrew, what you've shown there is actually reinforcing the whole knowledge driven product process development. So you're right, 25 years ago, we would throw everything that we could think of at a molecule to try and understand what it is. Now what we do is quite a structured approach, because we know what's going to work. So that's that knowledge-based product process development. But to my mind, Fiona, in terms of the quality by design, I mean, I've been in the industry long enough now to have seen things like parametric release, real-time, real release, quality by design, design space, and my strong feeling is that we're repackaging the existing frameworks to generate data, to encourage people to look at the product process holistically. We're repackaging that, but there's really nothing new, because it's all good science. And as good scientists, as our knowledge develops, we learn from that, and we adopt and implement that going forward. So yes, quality by design is a great concept, but if you look at the basics, Andrew's just really clearly shown a very systematic approach in development that's based on knowledge.
Yes, yeah, I agree with that, Ash. Yes, it is. It's all based on this strong foundation that you have at the beginning. I'm sorry, but in the interest of time, we're going to have to stop our discussion here. Thank you both for a very stimulating discussion, and I feel we could go on and talk more about this. I've got some more questions that I'd like to explore with you, particularly on the issue of qualification of methods that you've mentioned. So I think we will go on and talk for a second session. So there'll be a second video cast. But for the listeners, thank you for listening to this video cast, and to Biopharmaspec for hosting. And please look out for part 2 of this by visiting the Biopharmaspec webpage or following on LinkedIn. Thank you.